So a couple of people who kept good records of what it was like to be here in Australia in the mid-1800s. George Augustus Robinson was the chief protector of Aborigines in Victoria, but he had come from Van Diemen's Land from Tasmania, and his job was to basically try and stop Europeans from massacring Aboriginal people. But as part of that, he kept a diary every day. Every single day he wrote in a diary of what he saw on his travels. So he's, with a wagon and some horses and a few other people. And it was fascinating because his description of the Victorian landscape was absolutely nothing like the Victorian landscape is today. In the middle of summer, February, like his diaries were like 1840, 1841, 1842. 1840, February, it hasn't rained for 90 days. Temperatures have been over 100 degrees for 90 days. And he's looking at green, luxuriant grass in every direction. Don't laugh, somebody's laughing down the front there. This is not a comedy. And South Australia was exactly the same. In February, in the 1800s, like no rain for months and months and months, and still green grass. Remember all the cattle that were driven down from the Gulf country, came down through the middle of Australia, plenty of feed and water on the way. There's no feed and water there now, and they came to plenty of feed and water here in South Australia. That feed and that water is not there. This landscape is so totally changed. Now, George Augustus Robinson, his diaries, if somebody took the trouble to sit down and type it all up from his handwriting. Apparently it was incredibly hard to read because he filled the page and then he turned it upside down and filled back the other way because he didn't have, much paper to write on, and then after he'd filled it up the other way, then he did all around the edges, and then he turned around and went on to the back. So it would have been a fairly, time-consuming thing transcribing. But the information that's in there is extraordinary. It's one of my most precious manuals, is this, his diary. Luxuriant grass in the middle of summer, carpets of colourful wildflowers. Everywhere he looked, he talks about these huge patches of reds and oranges, yellows, blues, colours that we don't even see in the wildflowers now, some of them. The red ones must have been scarlet runner peas or something that I don't think even exist here anymore. The deep soft soil, you could push a stick in 2 feet into the ground easily. Now remember that the Aboriginal people were digging yam daisies and tubers off orchids and all these flowers that were in these grasslands as their staple food. There was plenty of food for people. There was abundant wildlife. There was lots of water and lots of life in that water, lots of life in the ground, on the ground, whatever. It was an incredibly... If we had to survive now in the landscape that we Europeans have created, we would die. We would starve if we went out there because there isn't any food out there anymore. And still green after 90 days with no rain. So the other evidence that we have is some of the paintings that were done in the 1800s. These are from the colonial landscape sections of the National Art Gallery in Canberra. I don't have any for South Australia. Has this got a... Oh, yeah. These ones are from Victoria. But these grasses that you're seeing in these paintings, so this is 1858. These are all native grasses. Okay, beautiful green, luxuriant native grasses that don't exist. Well, they're almost extinct. They do exist, but you don't see much of them. And this is middle of summer, so these are warm season grasses. And you'll notice there's not many trees. These trees that have been planted here are European trees. Hills don't have trees. George Augustus Robinson talked about all the hills not having trees. And it was an open landscape that people were able to just come in and start grazing straight away. Again, here's another one from 1860. These are all native grasses. They're green, they're luxurious. This was a hydrated, highly productive landscape. How come it was so green over summer? How come it didn't dry off? How come there weren't fires? Now, this is a photo from Colin Sizer's place, and Colin's going to be talking to you later today after me. These are a beautiful mix of mostly warm season native grasses and beautifully green. This one's, again, warm season native grasses. This is what much of your South Australian grasslands would have looked like at the time of European settlement. February 2010, this is native grass. Okay, warm season native grass. This is a Paspalodium, and again a close-up of it. People don't think of native grasses as being like that. This is the middle of summer. Warm season grasses have the ability to grow very well in summertime. Now, the other thing that I want to talk about is Sir Paul Edmund Strzelecki. He was a Polish explorer. He came here looking for gold. he also was very fond of the wives of some of very important people, governors, etc. And he had quite a reputation. He kept a diary as well, which unfortunately had a lot of information in it that he didn't want to have published. And his diaries, according to his will, were destroyed on his death. But there was a huge amount of ecological information and soils information in there. But fortunately, he had written some papers, and he just kept those to the science. And one of those was about the soil. And he had, in 1839, he collected 41 soil samples. He sent them back to Kew in England, and had them analysed. Now, the 10 highest ranking of those 41 samples had organic matter levels from 11% to 37.75%. These were soils that these were on farms, these were arable soils that farmers were growing oats and things for their horses. It wasn't out in the bush somewhere. It was these are farm soils. How many farm soils now have organic matter levels between 11 and 37.75%? And even the 10 lowest ranking soils had organic matter levels from 2.2 to 5, which we would consider these days to be quite good. Now, the interesting thing that he also found was that when you took the lowest ranking soils and compared their water holding capacity to the highest ranking soils, there was an 18-fold difference in soil water holding capacity. Okay, so we had green luxuriant grass all year, even here in South Australia. We had a hydrated landscape that didn't burn. We had soils that could hold massive amounts of water. The climate hasn't really changed. Well, it has changed a little bit because of many of the things that we've done. We've created microclimate effects, and I'm going to mention them in a moment. But we have changed the nature of our soil because we've changed the nature of the ground cover. And that's going to be my main message today. The organic carbon content of most of Australian topsoils, not just here in South Australia, but generally in Australia, is 50 to 80% less than the original level. And well, why does that matter? Well, I'm sure most of you are very aware because you've come through on your surveys saying that you know your organic matter levels are now low. Because the organic carbon content is the key driver for the nutritional status of plants. I won't have time to mention very much about that, but I'm sure you're all aware of that. And therefore the nutritional status of the animals and people. So if we have animals that have issues, it's going to come back to the soil. And why is the soil not functioning as it should? And people. We have huge metabolic issues in the human population now. There's an epidemic of autoimmune disorders. I've been a victim of it myself, and I've had to change my diet drastically. And it's around the world. It's because the food we're eating, the food that we're eating doesn't have the trace elements and things in it that it should, and that's because those things are in soil, not available.
You need to fill it up with electricity.
They're not available to us because they're not in the plants, because the plants aren't able to get them from the soil, because the microbes that we need in the soil to make those things plant available are not there, and those microbes are not there because the plants that they need for support are not there. The organic carbon content of soil is the key driver for moisture holding capacity, as I've just shown from Strzelecki's work, and that's frequently the most limiting factor for production. I'm sure you're all well aware of that. And for those reasons, because it's important for moisture holding and nutritional status, it is going to be the key driver for farm profit. So really we have to understand how to get more organic carbon in our soil, and unfortunately 30% of the world's cropland, so I'm talking generally now, globally, has been abandoned in the last 40 years due to soil decline, and that soil decline is as a result of losses of organic carbon, which affects its structure and everything else. So what's changed? Just let's go back to South Australia again. What has changed over time since, I'm talking about the time since European settlement? We've simplified the landscape, hugely simplified. Our original grasslands had 300, 400 species in them. How many species do you have in your paddocks now? I was just in England earlier this year, and a farmer told me very proudly that he had 200 species in his paddock, meadow grasses and forbs, herbs in his paddock, 200. They have up to 400 as well in a healthy grassland. Our healthy grasslands have 300 to 400. So we've simplified the landscape. Just flying over yesterday, I'm just looking down from the plane, and mostly what I'm seeing is farmland that's obviously had cereals in it that are in the process of being harvested. So we've reduced the amount of green, because how green is it now in February and March? January, July, summer months. From now on, it's not going to be very green. We've reduced the diversity, as I mentioned. The soil fertility has declined, and you've come up with that in your answers to the questions. Everyone's aware of that. Why are we using fertiliser? When crops were first grown here in Australia, we didn't need to use any fertiliser, and then over time people have been using more and more and more, and that's because soil fertility is declining, and it's now dry when it once was hydrated. So they're all of the things that have changed, and we can't consider plant soils, microbiology and hydrology in isolation. That's one of the other issues. We've got, people studying plants, people studying soils, people looking at microbiology. We need to see how the whole functions, how all of those... This microphone's going to drive me crazy. It's not me, it's this. It's not my voice, it's the microphone keeps cutting out. Okay, so if we look at this, just put the whole thing into one picture. Here, healthy soil is because it has green living ground cover year-round. Net primary production decreased, that means something has happened. Like in Australia, what happened? We came in with massive numbers of sheep, which increased very, very rapidly, like numbers were almost doubling. I think every 10 years, sheep numbers doubled in the 1800s, and we got overgrazing. Once you start losing ground cover, then you get soil degradation. Then you start losing soil organic carbon, and then you start losing moisture because the soil simply can't hold moisture. You get massive increases in evapotranspiration, you get more aridity, you get carbon dioxide emissions. A lot of the carbon dioxide in our atmosphere has actually come from soil. And this whole warming can come from losing green. How do we know that climate change isn't as a result of how we farm and how we have changed the landscape hugely? In fact, if we could get it back to being like this, I think we'd see a dramatic improvement in our climate. And if we look at that in terms of, we already see this in Australia. In Western Australia, for example, here's the outline of Western Australia here. The southwest of Western Australia is there. This is just a photo of the clouds. Now this red line is the rabbit-proof fence. I'm sure some of you will have seen these photos before of how there's moisture, there's cloud formation on the vegetated side of the rabbit-proof fence. There's no cloud formation on the farmed side of the rabbit-proof fence. Now, in this case, the vegetation is scrubland, but the reason that we've got cloud formation is because the moisture in the atmosphere is being nucleated by bacterial aerosols from that scrubland, and grasses produce bacterial aerosols as well. So it wouldn't matter whether it was scrub or whether it was grasses on that side of the fence, you would still get that. So if our farmland is this section here that's got no cloud over it, there's obviously sufficient moisture in the air to form cloud, but we don't have the bacterial aerosols nucleating those clouds if we don't have vegetation there. And it's not just about the dynamics of the atmospheric water cycle. We have to look at the dynamics of the water cycle in our soils as well. And we see that actively growing green plants support the microbes that create well-structured, friable topsoil with high nutrient status and high water holding capacity. And in a half an hour I'm going to try and explain to you how that happens. So if we had more time, I would ask you what this is a photo of. Some of you might have seen it before. In fact, there's a few repeat offenders here. I see some people that I have seen before at my workshops. So you've probably seen this photo. But this is taken in the Western Districts of Victoria, near Horsham. And it's a photo of, obviously it's a wheat crop. And the farmer has just taken this fence line out to create one large paddock where he had two smaller paddocks. And it's in a drought year, and he was about to bale this for hay. So it's a failed crop, but the crop has not failed where the fence line was. Why? And this is no-till, so it's never been cultivated. It's not that the soil structure has changed through cultivation, or some people would say, okay, if you had offset discs, you'll get soil thrown up against the fence, and so it could have had deeper soil there or something. What you're seeing is that where the fence line was, there were plants growing year-round that no one bothered about, probably weeds. but there was something green there. Whenever it rained, there was something that could respond to that rain. There was something green, and those green plants were keeping microbes alive in the soil. So you had more microbially active soil, and it was able to support that crop. No problem at all. That this is not showing any effects of drought. We have created the drought by the way we farm. So it's not necessarily on continental scales or global scales as well. It works at the very small scale in your farm. And in fact, it works right down to the nanoparticle size scale almost, because we start talking about microbial hotspots around plant roots and what actually happens in those hotspots. And I'm going to get to that in a moment. So it's from the tiniest scale that we can imagine to statewide scales or regional scales, continental scales, global scales. All depends on what is actually happening around plant roots. And you are in control of what happens around the roots on your plants, because you are in control of what plants are there and how they're managed. The presence of green plants is the most important factor for soil health, and then obviously how you manage those plants. So how can we use what we've learned from history to rebuild healthy, porous, carbon-rich topsoil? Well, to answer that question, we first have to ask several others, and the first of those is actually, what is soil? If we had more time, if this was a workshop where we had time to explore this, we would spend a lot of time now actually talking about what soil is. And what amazes me is that there can be a room full of farmers, as we have today, if you don't mind me using that word. Some people don't like that word, farmers, but you know what I mean, people who produce something from soil who can't tell me what soil is. In fact, most people actually can't tell me what soil is, but we haven't got time to go into that day. And by what processes does it form? So I'm going to give you the answers. What is soil and where does it sit in the hierarchy of life? Well, how often have you heard the phrase that soil is the foundation of everything? Actually, put your hand up if you believe that. We've got time to be a little bit interactive, thank you. I used to say that myself. Soil is the foundation of everything because, it's under our feet, not so much today, but it's under this building, it's under our crops and our pastures and our livestock, and it's under everything. And we've got this idealised conceptual framework where we see soil, this is very simplistic of course, but you'll see this in almost any textbook you pick up on soils. It'll be a bit more, upmarket than this, but there'll be soil. And then there's plants, obviously they grow in soil, and then there's going to be animals because they eat plants. And we all think of it being like that. Yes? You all think of it being like that, in simplistic terms? And then the problem is that if our crops and pastures aren't performing as we'd like, we want to add something to the soil to fix it. So we go, our plants aren't performing as we'd like, or our animals aren't growing as quickly as we'd like. So what do? We go back, we take a soil test, we measure the soil, we say, it's missing this, this and this, or it's too high in this, this and this, and we want to start adding something to that soil to try and fix the problem. Well, what if soil is not the base of the pyramid of life? Have we been directing our energies to the wrong thing? And if it wasn't soil that was at the base, what should be at the base? So the definition of soil, it's weathered rock materials. I'm sure you all realise that. There's parent material somewhere that's broken down into sand, silt and clay, and you'll all have different proportions of that, and depending on where you are in the world, you'll have a different soil type. But it's not actually soil. Weathered rock material is not soil unless it has been in contact with plant roots. So if you go somewhere in the world where there aren't any plants, where it's too hot, too cold, too dry, too something or other, go to the top of a mountain above the tree line where there's no plants because it's too cold, and you'll find rocks up there and you'll find some weathered rock materials up there, but you won't find any topsoil up there. Go to a desert, the Sahara Desert or whatever, or inland parts of Australia and Simpson Desert, and if it's too hot and too dry and there's no plants, there's no soil, you'll find sand there. Okay, go to Antarctica, it's too cold, you'll find weathered rock materials there but no soil there. So wherever there are no plants, there is no soil. Where plants grow really well, there is good, deep soil. So there's this relationship between plant roots and soil formation, and specifically roots we're talking about. It's because green plants captures sunlight and carbon dioxide. So in this miracle of photosynthesis, these leaves of this green plant are going to take atmospheric CO2, convert it into sugars, and translocate it down through the root system of this grass here. And these roots go even deeper than what's shown in this photo. The roots of this grass have created this topsoil. They have created beautifully aggregated, porous, fertile, high water holding capacity, well-structured topsoil from what was just lifeless mineral soil from the breakdown of rocks. When rocks break down, they produce stuff that you wouldn't want to be growing crops in, although a lot of you are now, or growing pastures in, because the soil has lost all of the other constituents that it needs to make it well aggregated and porous and high in nutrient status. So plants are the only things on the planet that can do this. And we have to understand how they do that and how we can promote soil building from plants. And their roots. We have to look at, how deep are the roots, how fibrous are the roots, and what is actually happening around the roots. You need to look really carefully at the roots of your plants. You want to be looking for things like this. In fact, you want to be never really able to see the roots on your plants. If you dig them out carefully, this has just been yanked out, so some of these you can see the roots, but it should all be like this. If you can see the roots on your plants, you have a problem. The soil should be sticking around the roots, and when it's sticking around the roots like that, that's soil building, and I'm going to show you what actually is happening inside those. We call these riser sheaths, so it's like a column of soil around plant roots. And these are aggregates. These are little lumps of soil stuck to plant roots, and we're going to, I think the next slide, we're actually going to look inside one of these aggregates and see what's happening in there. I'm going to be going over this really, really quickly. But you can find this information on my website, and if you want to go back, if it's of interest to you, want to go back more slowly and look over it. So this blue circle here is a macro aggregate on a plant root. The green column down here, this is a fine feeder root of a plant. It's probably too fine for us to really see easily with the naked eye. These are root hairs, which often we cannot see with the naked eye. They come up really well on with a microscope, but difficult to see with the naked eye. These other little strands that run out from this plant root are the hyphae of mycorrhizal fungi. They're like little hairs running out. They go right into the plant root and then they go out into the soil. And they're pulling all those soil particles together to form that macro aggregate. But within that macro aggregate, there's these orange shapes, which are micro aggregates, these soil micro aggregates here, which are very small, and they're formed by bacterial glues and gums. So the sticky substances that the bacteria use to form these micro aggregates and the sticky substances that the mycorrhizal fungi use to pull it all together into a macro aggregate come from sugars. They're polysaccharides that are made from the sugars that were formed by the plant in photosynthesis. So unless you have an actively photosynthesizing green plant making those sugars, translocating them to the roots and having them being utilised by the bacteria and the fungi in the root zone, you're not going to have soil sticking around the plant roots. So why is it important that we have soil sticking around the plant roots? And that's because the reason it's important is because inside those macro aggregates or inside the riser sheaths, which are like columns of soil on the outside of the plants, we have an energy-rich environment, but also an environment that's very low in oxygen. It's got a very low partial pressure of oxygen, and it's very, very similar to the environment that's inside a nodule on a clover or some other kind of legume. I'm sure you're all familiar with nitrogen fixing in legumes. So there's rhizobia bacteria inside a nodule on the roots of a legume. If you squish it, like with your thumbnail or something, you'll notice that it's very moist inside, and also it should be a pinkish colour, and that will tell you that nitrogen fixing is taking place. Now, a nodule on a legume has a low partial pressure of oxygen, has a high moisture content, and has a high sugar content. The same happens in an aggregate, and the same happens in a riser sheath on plant roots. Why low oxygen content? Because the enzyme that nitrogen fixing bacteria use to fix nitrogen from the air is 78% nitrogen. That means there's 78,000 tonnes of nitrogen gas sitting above every hectare of your land. Your soil should never ever be short of nitrogen. These little yellow ellipses here are colonies of nitrogen fixing bacteria. They get their energy from the sugars that come out of these roots. and they produce an enzyme called nitrogenase that converts atmospheric nitrogen into plant-available forms of nitrogen. They can't do that unless they're in that protected environment. So if your plants aren't forming rhizosheaths and they're not forming aggregates, you're not getting enough nitrogen, so you'll be tempted to add nitrogen because plants need nitrogen to grow, and once you start adding nitrogen, then you throw the whole soil ecosystem out of balance. So we have to understand how these processes work. I'm sorry if I've gone through that too quickly, but on my website there's a paper called Nitrogen, the Double-Edged Sword, which has this diagram and explains it all over several pages as how it actually works. The reason that this is so important is because what is formed inside an aggregate when we have nitrogen-fixing bacteria, there's also phosphorus-solubilising bacteria in there, sulfur-fixing bacteria, and bacteria and fungi that are utilising carbon, is that all of those things are polymerised into humus. This is the holy grail, is to form humic substances in soil from simple sugars that come out of plants. What's humus made of? Well, it's about 60% carbon, 6 to 8% nitrogen, 1 to 2% phosphorus, and 0.8 to 1.5% sulfur. Again, you'll find all this on my website. If you add those things together, it comes to about 70%. So there's another 30% of other things in humus. What is the other 30%? The other 30% are minerals. Things like aluminium and iron that get taken out of solution and sequestered into the humic molecules so that they're no longer toxic to plants. So if you have issues with soils that are high in available aluminium or high in available iron, it's probably because humification is not proceeding, or humus molecules are breaking down and releasing the iron that's in there. So it's an organo-mineral complex, it's part of the soil matrix, it can't be extracted from soil. If anyone tells you they're extracting humus from soil, they're not. You can extract humic acids and fulvic acids, but you can't extract humus because it's a polymer that is part of the soil. In the same way that trees photosynthesize in their green leaves and they produce sugar in their green leaves, and it's translocated in the sap throughout the tree, and the tree polymerises that carbon, those simple carbon compounds, to make wood. Lignification is a very similar process to humification, and once it's wood, it's wood. You can't extract wood from wood. It's either wood or it isn't. And it's either humus or it isn't. We can extract tannic acid from wood, we can extract humic acid from soils, but we can't extract humus. Just want to make that clear because there's a lot of misinformation about humus in the literature. What does it look like? Well, it's a gel-like substance. It's pretty colourless. It's a bit like crude oil. It's probably, if it's got a colour, it's honey-coloured. When we see organic matter breaking down in soil, we see darker coloured soil near the soil surface. That's not humus. And I'm going to explain in a minute about the differences between humus and organic matter. This is a different thing altogether. It's formed of carbon atoms that form hexagons. So they've got 6 carbon atoms forming, they're called rings, but they're actually hexagons, and they're all linked in a lattice. So once you actually get this structure formed, it's very stable in soil, can be there for thousands of years in fact. And it has a very high cation exchange capacity because there's lots of sites here that other nutrients and things can link to, so it will hold nutrients in your soil. It's got very high water holding capacity. It's more important than clay for holding moisture in your soil, and it's more important for clay for holding nutrients. It's got a higher cation exchange capacity than clay. This is the holy grail. If you've got sand, you can build it with humus, okay? But you need to understand how to get that humic molecule in there. So what I'm talking about basically is the liquid carbon pathway. I'm talking about sugars being formed in photosynthesis. They're in the sap of the plant. Some of them are used to build the plant itself. Some of them are polymerised to make cellulose and lignin and those kinds of things, the above part structures of the plants, and to produce flowers and seeds. Some of it's translocated to roots. And then if we have soil that's functioning effectively, we will have aggregates formed around those roots and humification take place inside those aggregates. Remember, humification can only take place in a protected atmosphere or a protected microsite where there's a low partial pressure of oxygen. It can never take place anywhere near the surface of the soil, and in fact the deeper in the soil that you can get your plant roots, the more likely it is that there'll be humus formed around those roots, because humification occurs more easily the deeper in the soil that you are. The closer to the surface there's too much oxygen, plus The hyphae of mycorrhizal fungi are very sweet. They're like candy floss, and mites and other things that live near the surface of the soil like to chomp on them, so it's pretty hard for mycorrhizal fungi to exist near the surface of the soil. So this is the liquid carbon pathway. It has 4 steps. You must have photosynthesis. Can't work without that. You must have translocation to roots. If you use lots of inorganic fertiliser, high rates of N or high rates of P will actually stop this from happening. You won't get translocation to the roots. They will definitely stop aggregate formation and humification. So what we've done in the way that we've simplified landscapes, we've gone from not having green all year round, we've gone from maybe one crop where we had hundreds of different kinds of plants before, and these steps aren't happening anymore. We're wondering why our soils are going backwards. So if we want to rebuild soils, we have to figure out how to reinvigorate that whole thing. So the first 2 steps here, photosynthesis and translocation, obviously require actively growing green plants. And then the steps 3 and 4 require a diversity of beneficial soil microbes, not just one kind. We have to have a whole lot of different functional groups. And the most recent science that's coming out is showing it's not so much to do with how many microbes you have in soil, it's not to do with microbial biomass, it's to do with that. Have you got the key functional groups and networking with each other and working together to actually build humus? You could add organic matter to soil and have zillions of, like put manure or something into soil and have zillions of microbes there, but they're not building soil because you haven't got the key functional groups that need to work together.
Question. If you've got aggregation around the root zone of your plants, could you make the assumption that you have humification happening, or is it something you can actually test to determine presence or absence?
That's a very good question, but if you've got aggregation happening around your plants, you've got soil carbon increasing around your plant roots. Definitely. I mean, because the aggregates are formed with sticky substances that are pulling the soil particles together. I'm going to show you a photo in a minute of what it actually looks like, and then you'll get a better idea. It can't, if you've got aggregation happening around your plants, there's got to be more carbon there because that's what the aggregates are made of. They're actually soil particles stuck together with carbon compounds. So humus-rich topsoil is a product of photosynthesis and microbial resynthesis. That's what I was getting to. And it's photosynthesis, not soil, that forms the base of the pyramid of life. So we have actually been looking at it upside down. Because if we want soil and we want fertile, productive, well-structured topsoil that's going to do for us what we want soil to do, then we can't have it unless we have photosynthesis, which is just green plants, light, carbon dioxide and water, and good. We're going to talk more about plant roots, and maybe Graham and Cole might talk about roots as well, and their associated microbes. You know, if we don't have the microbes there, we're still not going to get soil. But so just imagine that this is really a very tiny part of this pyramid. Photosynthesis is huge, and if we start reducing that, like we're seeing in South Australia at the moment, where at least we're cutting that in half because there's not a lot of green stuff on the ground around here over summer months, then immediately you've cut your soil formation in half. And then with the photosynthesis that we've cut in half by having it only happening here in wintertime, and we've got a really limited number of plants that are involved in photosynthesising, then we're making this even smaller. So now we're working on this tiny little fraction of maybe a bit of soil formation. And in actual fact, most of you, I'm sure, are not meaning to be challenging or in your face, are not actually forming topsoil on your farms. Your soils are going backwards, which is why you're here, losing topsoil. So the word photosynthesis, photo means light, synthesis means putting together, and that word photosynthesis literally means making life from light. So what do the roots look like when the liquid carbon pathway is not functioning? I thought I'd just throw a few of these photos up just to show you quickly. This wheat plant here on the right, that's the seed there. It's had 100 kilos per hectare of MAP put underneath the seed, and this is generally what you'll see in most wheat crops in Australia. You'll see short, dysfunctional root system with no aggregation happening around the roots, no riser sheaths, and this poor thing has got, like, there's no life here under this plant, and what happens when the topsoil dries out? Well, it can't even get water, and there's certainly no soil forming happening under this plant. These two plants here, the same age, same variety of wheat, pulled out from about a metre apart, away from this. This is from a research trial where we're looking at pasture cropping. So these two wheat plants here are grown in a pasture crop soil. again, they were just pulled out because we weren't thinking of taking the photo, and then we had them and said, oh, let's take the photo. So some of them look clean, but they would have actually all been covered. These have got rhizosheaths on them. They sort of look like dreadlocks. And if I've got time, I'll show you some more photos of plants that are building soil. Much deeper root system. You can't see any of these roots down here. So under these plants, we have soil building occurring at depth. Under this plant, we have soil decline, and we'll end up with a hard pan here, whether we cultivate or not. We're going to end up with a line here where water can't even get down. So then when it rains, if water can't get down to here, and this soil can never get hydrated. You know, I think this photo describes really well how plants build soil and how now we're going to be able to store water down here because it's going to be able to get down there. So people say plants take from the soil, nothing could be further from the truth, and plants are going to dry our landscapes out, nothing could be further than the truth. You won't have a hydrated landscape unless you have plants.
Hang on.
Does soil pH set limits on humification?
Humification changes soil pH.
Right.
So once you get to the stage where you've got highly alkaline or highly acidic soils, it's harder for plants to grow, but getting some kind of plant that's going to be tolerant of whatever that pH condition is your secret to getting back to. We find alkaline soils become neutral and acid soils become neutral once we get humification happening, because the only way that you can really alkalise or neutralise, I should say, soils is through this humification process, because this will always bring the pH back to around about 6.5 to 7.
Can you go back to that photo? Are you looking at soil disease levels as well? Because that plant on the right, I'm just seeing, I know there's other issues, but I'm seeing rhizome through those root systems, whereas the plants on the left do not.
Well, disease is going to be a huge factor. That's not the reason, like in this case, like if you dig up wheat almost anywhere in Australia, you'll see a root system like this on an annual plant. Well, and it should be like this. But when you've got all this life, when you've got all these rhizosheaths and aggregation and everything happening around plant roots, that's teeming with life. There are trillions, literally trillions of microbes per gram of soil there, and virtually there'll be hundreds per gram there and trillions per gram here. A lot of these microbes that live around plant roots are very protective of the plant because that's their host. It's that plant that's providing the sugars and other carbon compounds that are keeping those things alive, and they defend the plant, so that when you have this happening, it's very unlikely that you'll get any disease in the plant. When you've got naked roots trying to survive without the help of microbes, microbes are incredibly important for protecting plant roots from disease. You want more microbes, and you want as many different kinds of microbes as possible, because it's diversity in the soil microbiome that actually provides the protection from disease. And we see exactly the same thing in the human situation. If you go to a hospital where you've got an operating theatre that's been sterilised, what happens in the operating theatre? You get golden staph. You won't get it if you had an operation on your kitchen table. You might die of something else, but you won't get golden staph. But, you know, the reason that golden staph is thriving in operating theatres, in Armidale, our operating theatre at our hospitals it gets stripped every 6 months. They take everything out. And it's all stainless steel and everything's all autoclaved, and it's kept too sterile, really. And golden staph can survive in that environment because there's no microbial antagonism. If you look in another environment, like in your home, there's so many different microbes there that golden staph can't survive. But then once you get it in your body, you can't get rid of it. It's the same thing in soil. We've sterilised soil in a way by not having enough microbial diversity there, and then the pathogens can take over. But we can discuss that more later this afternoon, because it's important now that I try and cover as much as possible.
Absolutely.
But this is, you're seeing lack of microbial diversity. If you're seeing root-borne diseases, you're seeing lack of microbial diversity.
And that is the problem with diseases like rhizoctonia.
Exactly.
It can dominate, but it's a very bad competitor if you've got the right microbes in the soil. Or if you can develop the right microbes in the soil.
We can talk about this afternoon, because, we could spend a lot of time talking about it, but microbial antagonism is the answer to soil-borne diseases. And to get microbial antagonism, you need microbes from a range of functional groups, which means you have to have a lot of different kinds of plants there. You can't deal with it in monoculture, really. Okay, so again, here we have clean roots. Like, if you can see white roots like that in your soil, there's something seriously wrong. And again, wheat, pickled seed. Now we've got, fungicide on the seed, or insecticides on the seed. This is not really good for a seed. We're just trying to establish relationships with things in the soil. We've got completely white roots. We've got absolutely no soil structure there. And, you can't even buy seed, as many of you would know, that hasn't got some nasty chemical on it. Why are we putting nasty chemicals on seed? Because we're trying to overcome soil-borne diseases or seed-borne diseases that result from lack of microbial diversity. So, anyway, I made the point. Prior to 1940, the importance of life in the soil was clearly recognised, and even here in Australia, CSIRO scientists were undertaking research into mycorrhizal fungi, free-living nitrogen-fixing bacteria. 1890s, papers coming out here in Australia on these things were regarded as being extremely important for soil. Hopefully we'll see a swing back to that. And those microbial groups are plant dependent. They can't be there in soil unless you have green plants. And in fact, most soil microbes are plant dependent. Back in 1938, most of the good soil information on soil biology came out prior to World War II. There can be no life without soil and no soil without life. They have evolved together. So when I'm talking about soils, you notice I haven't said anything about physics or chemistry or what your soil texture is or what your soil type is. It's all about plants, about diversity of plants and how your plants are interacting with your soil, because your plants are going to create the environment for life, and it is life that is going to make your soil. Why is liquid carbon different from just having organic matter in your soil? I'm not talking about organic matter. Okay, I am not talking about organic matter. There's two pathways for carbon in soil. There's the well-known decomposition pathway, which is the one that's based on organic matter that decomposes in soil, and the less well-known liquid carbon pathway, although this is now becoming more well-known. In fact, I googled it the other day and I found there was even some scientific papers where they've actually used that, and they use the acronym LCP, the LCP, and I thought, yes, it's taken about 20 years, but we're getting there. So the decomposition pathway ends in CO2. Every year I get some lucerne hay and put it around my roses and everything looks great for about 3 months or something, and then one day I walk out and go, oh, all that straw's gone, all that mulch I put on that soil has gone. Where did it go? It's all gone back to the atmosphere as CO2. Organic matter doesn't build soil, and now there's some really good science showing organic matter doesn't form permanent improvements to your soil. The liquid carbon pathway is the only way that we can build humus in soil, and I'm glad to see there's peer-reviewed science now showing that. So the decomposition pathway does start with photosynthesis. That's how we get the leaves and the roots and whatever it is that you use to make the hay or the leaf litter. It does produce biomass, but it decomposes and it turns back to be CO2. How many papers will you read that say, if you want to build soil carbon, you have to have lots of biomass? Yes, you have to have biomass for liquid carbon, but not for organic matter. So this is organic matter decomposing in the soil. There's the saprophytes, saprophytic fungi, that are forming this white mycelium. That's very beneficial. I'm not saying there's anything wrong with that. If you see that white material in your soil, that's great. These are decomposer fungi. Excellent for aerating the soil and feeding the soil food web. It's all good. Nothing wrong with it, but it won't stay there. Again, this is like lots of organic matter in the soil. Great for the surface. But what we're talking about here with organic matter is, like this is pretty much sand. This is from Baralaba in central Queensland on a cattle track where the cattle have walked all the time, and therefore there's no green stuff growing there. So that's what their mineral soil looks like. That's how it's improved when it's got organic matter in it. But if that is just left without continual additions of organic matter, it will go back to being that, because there is no way that that can be stabilised. It's beneficial, it buffers the soil from high and low temperatures, it maintains moisture, but we're just talking about the top couple of centimetres of soil when we're talking about organic matter, or at the most we're talking about maybe 20 centimetres down. We're not talking about deep. We want to be building carbon 20, 30, 40, 50 centimetres at a metre. We want to be getting plant roots down there and building soil carbon down there. So what are the most important principles for building deep topsoil? Because we want to get moisture way, way down where it's protected from evaporation and where it's going to maintain a hydrated landscape over summer. So you could grow summer crops here when you start to get hydration back into soil. It's going to be a gradual process of rebuilding soils, rebuilding soil depth, rebuilding soil structure, rebuilding water holding capacity. But you'll get enough moisture over winter for it to remain hydrated and for you to be able to grow plants over summer, because it was green here in summer, remember, not that long ago. So as to methods, there may be a million and then some, but it's the principles which are important. There are just a few key principles for rebuilding topsoil. And once you actually grasp the principles, you can select your own methods. You can be creative about this. In fact, I find around the world that it's the farmers that are coming up with the most creative ways of solving their problems and restoring their topsoils. An academic like me, we tend to be linear, reductionist type thinkers, and I'm just amazed when I go onto farms and I think, oh wow, look what they've done there. Because you're out there every day, you're looking at your plants, you're looking at your animals, looking at your soils and thinking about it, and you can come up with incredibly creative ways to do this. So I'm going to talk about the principles, and it's going to be up to you to come up with the methods, and hopefully today you'll come up with some of the steps that are going to get you from where you are now to where you need to be in terms of doing that. So it doesn't matter what you produce. I know we had a questionnaire, are you grazing, you've got livestock, are you growing? It doesn't matter what you are producing. You are first and foremost a light farmer. All of you are farming light, and you're just turning it into something else. And there are only 2 rules for light farming, so there's not many principles here that you have to worry about. You have to build photosynthetic capacity, and you have to enhance photosynthetic rate. Now, normally, because these are the fundamental principles for building topsoil, we would spend hours talking about these things. We haven't got time to do that this morning. I've got about 5 minutes left. So in that 5 minutes, I'm just going to have to race through this and give you some idea of what photosynthetic capacity might be. What do you think it might be? I can't ask you now, but you'll be thinking about it before I show you. Obviously, photosynthetic capacity is like, how much green leaf have I got there? I have to intercept solar radiation. I have to convert, my green plants have to convert light into carbon compounds. And then those green plants have to translocate those carbon compounds into the soil. So the photosynthetic capacity of any area of land is how much green leaf have I got there? What diversity of shapes of leaves have I got? Because we know if there's a big diversity in leaf shape, we're going to intercept a lot more light. If you've just got grasses, with the architecture of grasses, you're going to lose a hell of a lot of light that's not going to be intercepted. So you need a mixture of you need leaves that face the light in different ways, as well as your grass type plants. So photosynthetic capacity is basically how much green leaf have I got there and how much of the year have I got it there. Is it there for just a couple of months of the year, or is it there for 12 months of the year? So photosynthetic capacity, how much is there? Right. And I could go to no-till, so I'm not cultivating anymore. How much is there? Okay, right, so you got it. Good. There is no photosynthetic capacity if we have bare ground. This could be the same thing. Why can't we have it covered with green like that? Pasture croppings. Cole's going to be talking about that later. There is absolutely no reason why it can't be green over summer, but we have all this fear about the green bridge. Oh my God, we can't have green stuff over summer that's going to carry disease from one year to the next. Well, it won't carry disease if you've got a diverse soil microbiome. As above, so below. We want to have a good root system, and that means we need a good top on our plants. This is a native grass, curly windmill grass, I'm not sure whether it would grow here, but it would be a good one to grow because I'm sure it would grow very well in South Australia. Just some quick photo from Western Australia, sandplain country, used to be beautiful grasslands, summertime now, no grasses there, stock being confinement fed. Some farmers putting in perennial grasses to stabilise the sand, stop it from blowing all the time, provide a little bit of feed for livestock. Just really quickly, let's have a look under one of these grasses and see what has it done to this sand. So if you can do it in sand, you can do it in anywhere. These poor things, they've gone for 5 months without a drop of rain, and that's been grazed 3 times, so it doesn't look brilliant. But let's take some soil cores underneath that plant. What does it look like under that plant? Well, you can see it's changed the colour of the sand, so it's doing something. This is Gatton panic. It's a subtropical grass from South Africa. This is Department of Ag, WA Department of Ag data. So this is the top 30 centimetres or top 12 inches if you're still thinking in inches from under Gatton panic. That's that grass that I just showed there. This is a warm season grass. It will grow here. So this is Benue, which is north of Geraldton in Western Australia. So what we see, if we look between the plants, in other words, we're in those rows between the plants compared to within the plants, if we take soil cores within the plants. Sorry, I'm rushing now. Organic carbon there's 4 times more carbon under the plants than between the plants, 3 times more available phosphorus. No fertiliser has been added at all. It's very low value farmland and it would cost more than the land was worth to put fertiliser under it. So all these warm season perennials in Western Australia are sown with no fertiliser. This increased availability of phosphorus has tripled, the availability of potassium has tripled, the availability of sulphur has tripled. pH has gone from 5.8 to 7.1, so there's the answer to your question about pH. As a result of having a green plant there. And there's more information about that, but I just want to skip over that. Just going to show you now just an example from another one from Western Australia, because we're talking about a very hot, dry summer and inhospitable landscape and low productivity soils. This is, so Ian and Diane at Royal Catcham are part of the Wheat Belt NRM, and the Wheat Belt NRM did a survey of 50 farms in the area to measure soil carbon. This is the neighbour's place over summer. You can see that the soil, like it's very light soil, the wheat yields are generally around 1 tonne per hectare or something or other like that. So the Wheat Belt NRM collected soil from about 50 farms that look like that, and luckily they also collected soil from Ian and Diane's place. Their place looks like this over summer. They just let the grasses grow because they've got sheep, and they figure, well, why would we spray it out? I mean, these fallows, they're not naturally clean like that. Like you say to the farmers, well, how often do you spray to keep a clean fallow? Well, probably 2 or 3 times every summer we spray it to keep it like that, because somebody has told them that you can't have any green plants over summer because it's bad for the soil, carries disease, uses water, all that nonsense. So this has got wheat stubble all through it still. It's exactly the same, just over the fence. So what did they find when they did this survey? This was soil samples collected by an independent scientist. They used the National Soil Carbon Research Program, the SCARP protocols developed by CSIRO. There was 5 replicates at each of 3 depths, so they measured 0 to 10, 10 to 20 and 20 to 30. And all of the differences they found between the Haggertys and the neighbour were statistically significant, which blew their socks off. They couldn't believe that there was so much difference. The percentage increase compared to the neighbour, the Haggertys have 41.5% more soil carbon, nearly 30% more soil nitrogen, total nitrogen. Now remember, carbon and nitrogen are linked. They move together. If you're losing carbon, you're losing nitrogen. If you're increasing carbon, you're increasing nitrogen. It cannot otherwise be, because they're in the same molecule. Organic carbon and organic nitrogen are linked together in a polymer. They're in the same molecule. And soil water holding capacity, that's what this stands for, 13% increase in soil water holding capacity when they've got something green over summer, which Department of Ag and the universities and everybody's telling them is going to rob their soil of moisture. Okay, they're improving their moisture holding. And the improvement in their soil carbon is more pronounced at depth. So as I said, they took 3 depths, 0 to 10, 10 to 20 and 20 to 30. Oh, sorry, I'm pressing the wrong buttons here. 37%, 40%, 54%. At the 20 to 30 centimetres, so we get down to like this far down into the soil, and we've got 54% more carbon there at depth. So it's coming from plant roots. It can't be building deeper in the soil. If it was a breakdown, and how much organic matter is there on the surface? Virtually nothing. Okay, we're looking at really, really low amounts of organic matter on the surface, but we're getting carbon built down here. What did the CSIRO and the Department of Ag and even the guy that collected the soil samples and everything said? We have got no idea what's going on. Doesn't really surprise me. According to the West Australian Department of Ag, you'd have to add 96 tonnes per hectare of organic matter to build increased carbon by 0.5%. So they're not adding anything like even a tonne of organic matter, and they build it by 40%. So yeah, go figure. So quite obviously the increase in soil carbon didn't come through the decomposition pathway. Now this is what the roots on their wheat look like. This is the Haggerty's wheat. Look at this. This seed is just forming a coleoptile, hasn't even got green leaves yet, and look at its root. You can't see the roots. Okay, fantastic riser sheaths, beautiful dreadlocks, heaps and heaps of nitrogen fixing going on here. They don't use any nitrogen. They don't use any synthetic fertiliser. And their mycorrhizal colonisation is 73% at the 2-leaf stage. Like this little guy hasn't even got 2 leaves yet, but I can tell by looking at that he's got huge mycorrhizal colonisation on the roots. The labs can't believe it. When the samples come in, they go, we can't believe you've got that much mycorrhiza. now this is I said I was going to show you some photos. This is what it actually looks like if you use a high-powered microscope to look at what is going on around these plant roots. What you see is mostly sand. These are sand grains here, very, very, low fertile soil, but all these little things that look like cobwebs here, sticky stuff like spider webs or something, it's almost like, it almost looks sticky, doesn't it? Like pulling all the soil particles together and forming these environments, these micro-environments in the soil where humification can take place. That's what plant roots look like normally, like under conventionally fertilised farming. So I can see I'm going to have to stop here. Yeah, okay, so... I'm just going to have to flick through all that, and I'm just going to show you one more thing before so much I would have liked to have told you. Sorry about this. I think I spent too long explaining stuff. Look, this is one of the drought benefits. Look, this is triticale monoculture, and that's a cocktail crop right next to it. This is like droughted, and that's not. It's just like, that's monoculture, right? Just incredible. Anyway, this experiment in Germany, the Jena experiment's been running for 15 years. Got amazing results on diversity. They compared having one plant or 2 or 4 or 8 or 16 different plant species in these plots that they did. They had 78 different plots with all these different mixtures, and they looked at none, 100 or 200 kilos of nitrogen per hectare per year, and it was a multifactorial experiment. So you have one kind of plant with 200 kilos and one kind of plant with 100 and whatever, or 16 kinds of plants with none. You get it? Like all combinations of all of those factors. And they found that having 8 or 16 plant species produce more biomass. So if you're graziers, this is what you need to know. 8 or 16 plant species produce more biomass than having 1 or 2 plant species there with 200 kilos of N. So diversity did far more for their soil and their productivity than nitrogen fertiliser. So high diversity produced greater plant yield than high N. I'll leave it there. I won't go into any more detail on that. Okay, thank you.